Site-specific recombination molecular mechanism. So, as understood, recombination is a molecular process that involves rearrangement of genes. And in this particular session, we are going to look at what is the molecular mechanism of recombination taking place only at a very specific site. So, let us look at the learning outcomes of this session. So site-specific recombination is generally conserved as the recombination is between defined sequence elements. That means if there is a gene rearrangement that has to happen, it will not happen randomly anywhere. It will happen only at a specific site, and that is why it is considered to be conserved. The process involves enzymes. We again know that enzymes are very highly specific, and therefore recognition of sequence elements by the enzymes make it site specific. And it also involves a protein machinery that enables forming a synaptic complex like the Holiday junction. Enzymes named recombinases are responsible for cleavage and joining of DNA segments, which means just like the DNA topoisomerases, even recombinases are able to not just cleave but also join the DNA that it has cut. Such a sequential progress or process leads to the DNA segment either getting inverted or the DNA segment could be moved to a new site. Commonly, there are two families of site-specific recombinases. One that is serine recombinases. As the name suggests, the active site has a serine residue which takes part in the catalytic activity, and tyrosine recombinases have tyrosine as a part of or as an amino acid that is actively taking part in the catalytic activity. Now, let us first just understand what is the consequence of site-specific recombination. Say, for example, you consider you have a circular DNA and the circular DNA has recombination sites. Now, the recombinases using these recombination sites can carry out gene rearrangement in such a way that this portion, which is going to be rearranged, is inverted. This is one thing that can happen. The second thing that can happen is a DNA can get integrated into another DNA. So this is a typical example of how lambda phage DNA can get integrated within the host genome. And this happens because again of recombination sites. So it is these recombination sites that are recognized by specific enzymes that are called as recombinases. And the recombination sites are short, maybe around 20 base pairs or so. And these recombination sites are the sequences of which are conserved. And very importantly so, because only if the sequences are conserved will the recombinase actually recognize these sequences and come and bind to it. Now this lambda phage integration at the same site in the host genome. So it will not integrate anywhere else. It is going to integrate only at this site. That is because this recombination site that is present in the lambda phage and the recombination site that is present in the the bacteria, they have sequence homology and therefore they pair up together, and between the two you have the synaptic complex being formed, and the recombinase essentially using these recombination sites is integrating the lambda phage into the host phage. So this is something that is very important. So here what you observed was inversion. So the rearrangement is such that an existing gene is simply inverted, and in this case what is observed is clearly insertion. That means an exogenous DNA has got inserted in another DNA. So two recombination sequences are always present. So you can see here there are two, here there are two. You can see here there is one pair and here there is a second pair. So two recombination sequences are always present generally for recombination. One that enables the recombinase to bind, and the other one is mainly where the cleavage and the joining takes place. So therefore, for every site-specific recombination, you will always find that there are two recombination sites. Now let us look at what are the different types of conserved site-specific recombinations that are possible. So one that we already looked at when we looked at lambda DNA integrating into host genome is how you can have a DNA getting inserted into an existing DNA. So here you can see that this is one pair of recombination site, this is another pair of recombination site, and this region, the gray region that you see over here, is the site of crossover. So this X, Y, A and B are basically genes that are denoted. So when you have crossing over happening between these two regions, these are the two crossover sites, you can see how the entire between this crossover site and the other crossover site, you have this entire gene getting inserted. So what you very clearly observe now is that this crossover region, that is this sequence that is present on both sides of the integrated gene. And now you will have X and Y far apart, and you will have A, B present within the X and Y gene. And of course, the recombination sites are also present. Same way you can have deletion. So what got inserted can also get deleted. So again, using these recombination sites, the recombinase can come and bind and delete this portion from here, leading again to what was there earlier, that is the XY gene with the crossover region and the gene that was inserted with the, or the DNA that was inserted with the crossover region. So this is deletion and this is insertion. But like mentioned earlier, you can also have inversion. Again, you can see it is between two sets of recombination sites. So you only have the AB, which is present in this orientation, being present now in this orientation after recombination. So the type of CSSR, that means the type of conserved site-specific recombination, depends on the organization of the recombination sites. That is, how is the site or how are the sequences aligned. Now you can see that this red segments that is there, these are the ones that have moved. Okay, so you can see that from one place to the other it has moved, and again from this place it has moved to the other. Similarly, what you would also observe is that the orientation of the crossover region itself, if it is indirect repeats, okay, so here you have direct repeats. So in for deletion and insertion, the repeats that are present at the crossover is direct, while if an inversion has to happen, then the repeats that are present at the crossover region are indirect, or what is called as inverted repeats. So therefore, the orientation of these sequences decide whether it is going to be an insertion, whether it is going to be a deletion, or whether it is going to be an inversion. Now, looking at the recombination sites per se, as you can see that these recombination sites that are there, the sequences are within the sequences are interspersed a region that is called as a crossover region. So if you look at it in a higher resolution, you can very clearly see that the recombination sites are, these ones are inverted repeats, okay, and you have at the center the crossover region. So you can see TAGC, here too it is TAGC, but this TAGC is not a palindrome. And so basically what you understand is this region need not always be a palindrome, and therefore it is asymmetric. So the crossover region over here is definitely asymmetric. Now, these regions are the regions where you can have the recombinases coming and binding. So you can see very well that the recombinase comes and binds to each of the recombination sequence. Now, after the recombinases come and bind, you can see how the gene exchange takes place across the crossover site. So you can see how there has been a cleavage over here. Okay, there has been a cleavage over here, and this portion has shifted here, and this portion has shifted down. So you can see how there is an exchange between the two DNA, and this is aligned, or this is happening just because of the recombinases and the alignment of the recombination sequences. So this is how recombination actually takes place, and this is a general molecular mechanistic that is observed. Now, of course, we understand that there are two families of recombinases. So one of the families is serine recombinases. And as mentioned earlier, these recombinases have in their active site a serine residue, which with its OH, functional group as OH, is able to carry out the cleavage and the joining both. So as you can see over here, the OH acts as the nucleophile, and it is going to attack the phosphodiester bond in such a way that the OH of the serine is now in a link, or is now in a bond between the phosphate. So you can see how an intermediate is formed where the serine is directly bound to the phosphate, and this OH has been cut from this part of the DNA. So you can see how the DNA has been cleaved at this point. Now next what happens is this OH, which is now free from the phosphodiester bond, will again act as a nucleophile and cut it in such a way that the serine OH is removed, but what is formed again is the phosphodiester bond. Now understand that this DNA can be a different DNA. So when it cut the two, okay, it is giving rise to an opportunity for another DNA to come in and bind over here. So the cutting is of a particular DNA, but the joining may be between different two different DNA. But of course, with the OH of this DNA acting as a nucleophile and carrying out the activity. So eventually, what is important to note is that the serine of the active site of recombinase is able to cut, and it is also able to join. And when it is cutting, it remains bound to the phosphate. It is not releasing the phosphate. So this is an important thing to note. It is just same as DNA topoisomerases, where you have the phosphotyrosine being formed when the tyrosine attacks the phosphodiester bond and remains bound to the phosphorus. Now let us look at the entire mechanism by lieu of which the serine recombinases function. So you can see here you have the recombinase recognition sites and the crossover region in between. You must understand that when the recombinase is cut, you will always have a staggered end again. Only two nucleotides or one nucleotide which is having staggering, not more. Unlike what you observe in endonucleases, restriction endonuclease cuts. So the serine recombinases will cut in such a way that you would have a 3' OH formed over here, and the phosphate is bound to the recombinase. And the second recombinase also cuts, okay, at the crossover region, but you can see how there is a staggered cut. It is not a blunt cut, it is a staggered cut. But the phosphate is always bound to the recombinase per se, leaving the 3' OH free. Another important fact that you must understand over here is that the serine recombinases are cutting both the DNA strands. It's not one, but it is cutting both the strands of the DNA. So in both the DNA, this is the DNA, this is one DNA and this is the other DNA. So this could be donor DNA and this could be the target DNA. So here too you will have the recombinases coming and binding and cutting both the strands. So in therefore both the duplexes, you have a 3' OH generated and you have the phosphates attached to the recombinase per se. Now what obviously happens is there is swapping of the DNA that is happening. So you will find that this 3' OH is able to interact with this phosphate, okay, to form a phosphodiester bond, while the 3' OH of this is able to interact with the phosphate of R4. So you're practically looking at the fact that you have the R2 and the R3 going together and the R1 and the R4 going together. So the R2 is present over here, R3 moves here. So when the R3 moves here, you can see that the DNA is also moving. And same way the R1 moves down and associates with the R4. So you have a, a joining between the R4 containing DNA and the R1 attached DNA, and here you will have between the R2 attached DNA and the R3 attached DNA. So therefore now the joining is going to eventually lead to exchange of genes. Very clearly that you observe here, you can see that this is a specific combination, and now you have a recombination that has happened, and that's thanks to serine recombinase. And serine recombinase is allowing the mechanism to happen in a less number of steps, because both the DNA are being cut at the same time. So you would have this OH interacting with this phosphate and this OH interacting with this phosphate, and so you know that you have a holiday junction formed over here in this manner. Now we look at the second family of recombinase, that is the tyrosine recombinases. In case of tyrosine recombinases, the mechanism is slightly different, as the tyrosine recombinases do not cut both the strands at the same time. So tyrosine recombinases have a tyrosine residue present at the active site, and that too also has its OH group that is active. Now that OH is going to uh, bind to the phosphorus, and what you have formed is a 3' phosphorus tyrosine. So you get a phosphotyrosine formed, leaving a 5' OH. And you can see how in the two DNA, you have only one DNA getting cut at the first stage. Even in the target DNA, say for example, you have only one strand getting cut. Now, when you have this kind of a situation, you obviously know that the 5' OH will interact with the phosphate that is present over here, and the 5' OH that is formed over here is going to interact with the phosphate over here. So you can very well see how you already have a holiday junction structure, synaptic complex being formed. So this 5' OH is interacting with this phosphorus, and this 5' OH is interacting with this phosphorus to give rise to one holiday junction. Now what happens is, at the second recombination site where the R2 and the R4 are present, they can now cut the second strand. So the second strand is cut, okay? And with the second strand getting cut, you again have the R2 binding to phosphorus, forming the phosphotyrosine. The 5' OH is free over here. This is also going to bind to bind to phosphate end, forming phosphotyrosine, and the 5' OH is free. So this 5' OH will interact with this phosphorus, while this 5' OH will interact with this. So you can see you have another holiday junction formed over here. So in tyrosine recombinases, you will have two um, holiday junctions being formed between the two respective strands, and that happens one after the other because tyrosine is able to cut only one strand at a time. And of course, after joining, you can see how you have recombination happening, and at the crossover you can see you have patch recombination that is happening. So this is how recombination happens, but it is site specific. You can see that only if you have this recognition sites can recombinases come and bind, and when the recombinases come and bind, they cut at the crossover point. So therefore, this is how recombination by serine recombinases and tyrosine recombinases happen. So let us make the conclusions. Site-specific recombination can lead to three possibilities: inversion, insertion, and deletion of DNA, ultimately leading to gene rearrangement. The recombination sites are defined and organized to enable binding of recombinase, cleavage, and joining using a recombination sequence. Serine recombinases cut both the strands of the DNA and join such that there is exchange of genes, while tyrosine recombinases cut one strand at a time and enable gene exchanges, forming typical holiday junction structures. So conserved site-specific recombination enables integration of phages into host genome. They convert multiple connected genomes into single copies. They can regulate gene expression. They can help in cell division and carry out many more biological roles. Thank you.